Thank you very much, Mike, for agreeing to give a Build a Cell seminar, and you can get started.
Great, thanks a lot. Yeah, so I'm Mike Booth. I'm an associate professor in University College London, and we do a lot of work on nucleic acids and SNAke cells. So I'm going to talk to you about some of our recent work on remote controlling SNAke cells. So yes, so SNAke cells, we all have SNAke cells. That's why we're here, right? So SNAke cells, lots of different definitions. For me, it's maybe something with a cellular function that's in a minimal environment, right? So for a lot of our work, it tends to be lipid bilayer compartments, normally with transcription translation machinery, so DNA template, cell-free protein synthesis system, and then, yeah. So the reasons why I think they're super cool, they're completely customizable. You can put different types of bioparts, but more importantly, compared to living cells, you can put lots of synthetic parts, right? Which again, you'll see from my stuff is really important. They're not alive, which some people think is a bad thing. Is not necessarily a bad thing, right? You can use them in a very different way to living cells because they're not alive. They have a lot of less drawbacks because they're not alive, especially if you want to think about using them for things like therapeutics or bioreactors. So they've got all sorts of cool applications that a lot of people are doing in terms of studying living processes all the way to making therapeutics and sensing. And one thing that I kind of like always try and point out is I feel like SNAke cells fill this niche between living cell therapeutics and engineering and individual molecules, right? I think there's a really exciting position that SNAke cells can fill there. So cell-free protein synthesis, it's basically what I'm going to be talking about for the rest of it. And so I wanted to talk a little bit about it first and some of the things we've done to make our own. So cell-free protein synthesis, transcription translation, there's lots of different names for it. It's basically just doing transcription translation, but not in a cell, right? There's cool applications, including SNAke cells, obviously, but you need a lot of enzymes to do it, and the ribosome. And so to do it, you basically have to put DNA in a tube, ribosomes in a tube, energy mixes, amino acids, nucleotides, all sorts of tRNAs, which Kate's done a really cool paper on for that, and then transcription factors and translation factors, right? And this is a critical point, right? So people can get them from lysates or they can purify them. And so this is kind of how it's done at the minute. So you can get lysates, which are cheap, not very tailorable, not very pure, very quick. Or you can get pure express type systems, which are very expensive, very tailorable and pure, but take forever. And so one thing that I was super excited about when I read it, when it came out in 2019 from Sebastian Merkel's lab is OnePOT, right? Which is growing all these plasmids for the translation transcription factors all in one pot, and then just doing a quick purification, right? So the cost is much lower, but it still keeps the tailorability and purity, and the preparation time's less. So I really wanted to try it in the lab, right? So this is something that Jeff started in my lab, and then Charlie's taken it up. And so when we first did this, we got these plasmids for the OnePOT from Addgene, right? And we thought, well, they should be right? They're from Addgene. So when we first did a go through the whole thing, it didn't work at all, right? And so what Jeff managed to find out is that it's basically because of the plasmids, right? So we weren't getting some of these bands come up. We were running a gel of all the plasmids, but all the proteins expression, but we're getting lots that aren't there. And so then when Charlie started, what he did is he just whole genome sequenced all the plasmids, right? And so what you can see here, very obviously, is that all the PQE plasmids had mutations on them. And they all had the same mutations on them, which was fascinating, right? At the lac operator and the promoter, right? The same mutation was in all of these plasmids. And so that was obviously very worrying. So what Charlie did to start with was, well, can we make a PCR-based assay to just identify, can we find any clones that have the right sequence? And we found a couple that had the right sequence. And then when there were some from Addgene that we couldn't find any correct sequence in. So we had to then repair those using saturated mutagenesis, and we managed to do that. But the most important thing Charlie did was then putting them into bacterial strains which were stable, which had the lag repressor on the genome instead of on a plasmid. And so now, as you can see in this lovely gel, they now all express all of the bands which you would expect to see from the complete reference that's in the paper. So that's great. And so now put it all together and we can get protein, right? So the one-pot expression works similarly to Pure Express, which is fantastic, right? This is really great because it can allow us to make a lot more SNAK cells. So this is how we make SNAK cells. Lots of people do it differently. So we use phase transfer emulsion. So we make an emulsion of the cell-free protein synthesis system in lipid-containing oil. We then make a phase transfer column with lipid oil and an aqueous outer solution. And then we just centrifuge through, right? And we get diameters of roughly cell size. And so just to wrap up this one-pot part, for me, the most exciting part about one-pot is that basically we can get cheaper pure cell-free protein synthesis, which allows us to then make more SNAK cells per experiment, right? Much, much cheaper. And hopefully this will allow us to do much bigger and better things, both in the lab, but also, as a field. So great, SNAK cells, cell-free protein synthesis explained, right? So, but the rest of the talk is going to be focused on this other area of SNAK cells, which is much less well explored, which is trying to control their activity, right? So we really need to better control the activity of SNAK cells, right? Things like targeting their activity, the safety and toxicity, if we actually wanted to use them in any living system, and programmability, if we want to use them for computing or for interfaces with computers. And so the way we get control in these systems is chemistry, right? So we're a chemistry group as well as a synthetic biology group. And we tend to do a lot of modification on the template of the DNA that we put into the cell-free protein synthesis systems. So the first stuff we ended up working on was on light. So light's really cool because you get amazing spatial precision, biocompatible. Also, you get this idea of multiple wavelength orthogonality, which basically just means that you've got different colors, so you can activate different things with different colors. I mean, no one had shown that until we showed it, but it's a really cool thing. Tissue penetration isn't great with light, but with longer wavelengths, you can start to think about that, and that's something we're working on. So to get the control, we tried to do something really simple, which is just block a promoter from a polymerase binding. Right, so we just attached loads of streptavidin through biotins to a promoter through a photocleavable link. Because this is just a molecule that if you shine light on it, will break up, right? So what happens is you just break this bond when you shine light on the system, and it cleaves back to the DNA. And then you get your promoter unblocked and you get transcription translation. And we use very, very simple chemistry for this. So you can buy these amino groups coming out of the major groove in this position, so you still get the Watson-Crick-Franklin base pairing. In case of this modifier, right, you can actually buy this one with a biotin and an amine reactive group, so you can get this just done straight away in one step, and you've got your photocleavable promoter. This blue molecule we made ourselves in the lab, but it's basically the same idea, but it cleaves instead of with blue light. So to give you an idea, so this is work from Dennis, who made the blue light activated DNA. So we have a promoter that is now blue activated. So what you can see is amine DNA constitutes like the full cleavage, full photocleavage as a positive control. When we have blue DNA, we then bind the streptavidin, and that increases the size because we've now got lots of streptavidin attached to the promoter, so it's obviously much bigger. But then we shine blue light on it in this case, and it cleaves back, right? So that all of those streptavidins are falling off when we shine blue light on the system. And so when we put both of these blue and UV light activated systems together, we can get this orthogonal activation, where with the amine DNA, it always expresses, but now with the blue DNA, it only expresses when you shine blue light, not no light or UV light. And the UV DNA only activates when we shine UV light and not blue light or no light. So we get this orthogonal activity, and this is something that had never been seen before with either SNAK cells or with nucleic acids. So the cool thing is then we can start thinking about biocomputing and patterning. So we made an AND gate with light as the inputs. So in this case, we used split beta-galactosidase. So in that case, you've got alpha unit and an omega unit. You can encode those on two different DNA that's controlled with two different colors of light. And so only when we shine both wavelengths of light will we get expression of both parts of the beta-gal and then activation and output of the fluorescent product. So this is exactly what we see here. Only when we shine both wavelengths of light do we get this activity. And then the cool thing about that is obviously you can encapsulate that into a synthetic cell and get synthetic cell computing, right? So in this case, we've again got that AND gate. We've got the UV alpha, so main alpha unit, and then the blue omega unit, and then that will only make it when we're showing both lengths of light. And the really cool thing about that, which is something you can only do with light because of the orthogonal wavelengths, is you can do dual wavelength patterning, right? So this means that only where two wavelengths of light overlap can you get activation of the system, right? So this dual wavelength patterning gets really precise regulation. So Jack and Dennis in the group wanted to then do an off switch. So we had this on switch, could we do an off switch as well? And so for that, we used antisense oligonucleotides, which are just short DNA oligos, which if they bind to an mRNA, if you have RNase H present, which is endogenous in cells, it's how it's normally used in therapeutics, will then cleave the RNA, right? And that obviously then turns off the RNA from translation. And so we just put the same chemistry, light-activated biotin and streptavidin, on an antisense oligo. And what you can see here is that with the mRNA by itself, it's a nice single band. With an ASO, it just cleaves at that one site and you get the two bands. But with the light-activated DNA, without light, you get no cleavage. But now with light, you get this activation of a degradation, right? So turning on the degradation of the mRNA. So then if we could put those two things together, we can get a blue template that expresses a fluorescent protein that then gets degraded by an ASO that's activated with UV light. And so what that looks like on the mRNA level is if we don't shine any wavelengths of light, then we get nothing. But if we shine blue light, then we get transcription. And then if we don't do anything, it just stays transcribing. But if we shine UV light after 3 hours, then we get the degradation, right? So we've got an on switch and then an off switch, and all with light. And we can look at that at the protein level as well. So if we shine UV light at the beginning, we get this knockdown effect, right? Because we're degrading all the mRNA that we're making. If we shine UV light at the end, then obviously we've already made the protein. We can degrade the mRNA, but we're not degrading the protein. But what we can do is then we can tailor the amount of protein we get by shining the UV light and turning off the translation at different times. Okay, so we had an orthogonal on switch, we had an on and off switch. So we thought, well, let's do an orthogonal off switch as well. So we made two different ASOs that deactivated either the mRNA of M. venus or the mRNA of M. cherry. And so only when we got the right pairs should we get this cleavage. So here you can see complicated gel. Don't worry, just look at these last 4 lanes basically. Only when we have the correct pairs do we get cleavage. So only when we've got M. venus, mRNA and UV, and only when we've got M. cherry, RNA and blue do we get cleavage, not in the other systems, showing this orthogonality. And then looking at the protein level again. So now, again, we're only getting this knockdown when we've got the correct pairs. So we've got M. cherry and blue and M. venus and UV, not in the other situations. Okay, so a lot of that, some of that was in synthetic cells, some of that was cell-free. Then we wanted to see what we could push with the light-activated synthetic cells. This is work from Jeff. So we put cell-free expression systems in synthetic cells. These are now lipid compartments, not just lipids and oil. Lipid bilayers, sorry, in water, not just in oil. And we've got a fluorescent dextran often here, just so you can see the synthetic cells, and then we're expressing a fluorescent protein in this case. And so you can see with no DNA, just like in bulk, this works really beautifully. So in no DNA, you don't have any expression. In amine DNA, you have expression. But with the light-activated DNA, now you've got this great off to on switch. So you get very little expression, if any, without light, and then a turning on of expression with light. And so obviously with light, the cool thing is you can pattern. And so what we end up doing is encapsulating synthetic cells in agarose and then putting photomasks over, so showing that we can get really precise regulation. So each of these dots is a synthetic cell we're activating within the pattern, so we can get really precise patterning. Obviously with really funny patterns. So here's just a set of cards, spades, diamonds, hearts, and clubs. You can see pretty good patterning of all of these shapes just from photomasks of synthetic cells on top of the synthetic cells. So that was cool, but what we really wanted to do is, can we get a communication between these non-living cells, a controlled communication between non-living cells and living cells? And so what we decided to do to start with was use quorum sensing. So it's used quite regularly in the synthetic cell literature because it's quite simple and because it's got membrane permeable molecules which do it. So the way it works in nature, in bacteria, is you have a synthetase that makes a homoserolactone small molecule that then goes into neighboring cells and then activates more of that synthetase with a regulator. So what we wanted to do is, can we then recapitulate that in SNANEC cells? So we wanted to have a sender SNANEC cell that could make in this case, BJI is the common sensing system that we use, BGI, BGR, and make this synthetase that will then make this IV homoserolactone, which is a membrane permeable molecule, from these two membrane impermeable reagents, so SAM and IV-CoA, which you can both buy. In that case, we would make a homoserolactone signal, but we can then put control of that based on the DNA template here. And then we'd also need a receiver bacteria cell. So in this case, we have a GFP signal when we have the homoserolactone present. So BJR will be transcribed and translated, but only activate the promoter when it's got the homoserolactone there. So we had to make these things. So we started with the receiver cell here. So in this case, we want to show that we can get GFP activation only when we have the homoserolactone present, right? So we have this system, as I explained before. And so when we first tested this system that we got from Kamal Hain's lab in the US, I should have had a citation there. And what you can see is that it does work, but there is a really poor dynamic range, and you can see there's a really bad off state. So we wondered what that would be. So we'd found in the literature that people had done directed evolution on these systems. So that's what Jeff went and did. So we did quite a lot of directed evolution to figure out what we could do to make it better. And we made it a lot better very quickly, right? So what you can see now is that we're getting a fantastic decrease of the off state, increase in the on state, and overall a really impressive increase in the range. But what you can see has happened is that, yes, we've got one site being modified, but we've also mutated the start codon, which is really surprising, right? Really strange. Why would it be mutating the start codon, right? And so what we ended up finding out, which I didn't actually know before, and I don't know how many people know, is that you can start translation from non-ATG start codons. There's a couple that work, but they work much worse. Obviously, as you'd imagine, right? So CTG is shown to have 100 times lower expression than ATG. Right? And so what we then did is, well, can we just then change the ribosome binding site to 100 times weaker ribosome binding site? So we did that, and we got identical activity, right, to show that it is this problem of having better dynamic range just by lowering the amount of protein, the BJR protein that you're making. Right, really fascinating stuff, I thought. Okay, so that was the receiver cell. Then we want to do the sender cell. So the sender cell is just the DNA that encodes for the BJI protein, and then incorporating these two small molecules to then make the IV homoserolactone. So what you can see is that we did that. Here's an example of it in tube, but we did it in SNANEC cells as well, and then just added that, the media from that basically onto the receiver cells that we'd made. We can see we get really great signal, even with a really large dilution of the pure express. So you can see we're getting a fantastic amount of IV homoserolactone being made. So then obviously we wanted to put these two things together. So we've got the sender cells, but now with light activated DNA, and the receiver cells that we'd made previously. So we can encapsulate those into one system together, where we've got the SNANEC cells and agarose just like before, and we're growing the bacteria on the top. And so you can see, if you look at just the bacteria with no DNA, they don't express GFP, right? Obviously not. With the light activated BGI DNA, without light, again, they don't express. We've got this really tight off state where there's no expression. But then when we shine light, we get this fantastic on state from these little microcolonies now all expressing GFP as they're growing. And you can see the SNANEC cells here are still stable after we've finished. Okay, so light. It's cool, right? It's very useful, especially for patterning. It's very useful in vitro systems and things like biocomputing. and controlling that sort of systems. But if you want to work in vivo, light is not very good, right? It doesn't go through the skin very well, right? There are certain places on the body that light can be useful. But what you want to think about is you want about stimuluses that will work in vivo. If you want, think about controllable snake cell therapeutics. And so one of those stimuluses that would work really well in vivo is magnetism, right? So magnetism is deeply tissue penetrating, is also biocompatible. And when we use it with anodes and nanoparticles, you can then do an alternating magnetic field, right? So it's already clinically approved. And so the way an alternating magnetic field works is if you have a nanoparticle and you put an alternating magnetic field on the system, it'll do a really local increase in heat just around the surface of the nanoparticle. And so that's what we're going to be using the thing. And the other thing about nanoparticles is it's obviously trackable by MRI as well, which is really cool. So you can think about imaging as well as activating. So I'm not an engineer, but there are a lot of people working on engineering AMF systems to use in the body. So things like large solenoid coils to put around parts of the body to control activity, pancake coils to work to get things like within the head, and spatial precision of activation within the head. So there's a lot of other work going on to get this to work in the clinic even better than it already is. We don't have a big old fancy system. We have this system we're using that has a solenoid coil within this here. So we have 18 turns of a solenoid coil that we just dip a tube into. Right. So this is work from Ellen, and what she managed to do was go, well, can we just put nucleic acids around the surface of this? This makes something called a spherical nucleic acid, which has been researched quite a lot. Characterized less well, but researched quite a lot. And so what we did is, well, can we attach a promoter region of a T7 promoter to these just through hybridization, right? So what people may or may not know is that a T7 promoter will only work if it's double-stranded, right? So we could make these inactive templates that had a single-stranded promoter and a double-stranded gene of interest, right? So this is just, in this case, it's just an M neon green fluorescent protein. But obviously, in principle, this could be anything here. So the idea was, in this case, the promoter is attached to here, so it won't transcribe or anything, and this template is inactive. But if we could put a magnetic field on the system, an alternating magnetic field, that would locally increase the temperature around the nanoparticle, denaturing all of the oligo that's attached to the nanoparticle, and that would then allow it to hybridize to that inactive template to activate it. Then we would then get cell-free protein synthesis from this template. So to do that, I mean, there's lots of nanoparticles you can buy from the literature, from the internet, but these are very poorly susceptible to magnetism. So we decided to make our own using help from collaborators and colleagues. And so Ellen made these beautiful nanoparticles, really great size distribution. She then silica coated them to put amines on the surface. So you get this fantastic silica encapsulation, which is again taking a method from one of my colleagues here at UCL who's now moved. What you can see with this is the nanoparticles have a really small diameter. When you silica coat them, they then get bigger, they get positively charged. And then what we did is we then put DBCO on here, so a click coupling agent, and then we attached the oligo through copper-free click chemistry. So you can obviously buy oligos that have things like azides on the end. And then this is the T7 promoter here. And then we just hybridize those two together and attach them to the anodes and nanoparticles. And what you can see is they get much bigger, obviously, because you've got loads of DNA on the surface, and they get more negative, obviously, because you've got lots of DNA on the surface, which is negative. One thing that we didn't fully realize until we did it is that most people don't purify their spherical nucleic acids. And this is a big problem because you have tons of electrostatically bound as well as covalently bound. And so Ellen had to make a whole new purification, which was basically using an electric field to pull off all of the oligo that was just electrostatically bound, and then just bringing that out of a agarose gel to then carry on for the next steps. Okay, so just to remind you all what we're doing, so we're getting a spherical nucleic acid where we've got an anodes and nanoparticle core with a silica coating with the DNA attached, and then this DNA is the T7 promoter top strand, which then we have an inactive DNA. And when we put an alternating magnetic field on the system, this induces a heat emission just around the nanoparticle, which then denatures it and allows it to bind to an inactive template and then activate cell-free expression, in this case, making an amnion green. And through a lot of optimization, it works really nicely, right? So in this case, this is the inactive template as our negative control and our full template. And what you can see with the spherical nucleic acids, so the SNAs, without a magnetic field, we get a really great off state, really minimal expression without a magnetic field. But with a magnetic field, we now get fantastic turning on of transcription translation. Right? So now this is just this off to on state, but instead of with light, it's with a magnetic field. One thing that's really important about this is that, again, one of the reasons why we made our own nanoparticles is it allowed us to use a AMF field strength and frequency that is under the FDA and EMA approved safety limit for using AMFs in the body, right? So a really important part of this work. So then we can obviously just put all of that into SNAC cells, just like we did with light systems. So again, so now we've got the SNAC cells with the fluorescent dextran again, we've got the inactive template and the SNAs. And so in this case, without a magnetic field and with the inactive template, obviously we get no expression. With the full template, we get expression, but now with the magnetic SNAs and the inactive templates, again, just like we saw in bulk, without a magnetic field, we get a really good off state. And with a magnetic field, we turn on the transcription translation. So this is now SNAC cells that are activated by a magnetic field. And so that's cool. And one of the advantages of it is you can then access molecules and SNAC cells that are inaccessible in any other way. And so you may disagree with me that this is a tissue mimic. It's just a black tube. Tissue phantoms and tissue mimics are a big area, and there's all sorts of different types of them. A lot of them involve hydrogels, but hydrogels are useless for us because they're all transparent and light goes straight through them, right? We used agarose for all the light activated stuff. But one thing that's really important to mimic is the hard opaqueness of tissue. And so this is what we did with a black tube, where only alternating magnetic field is getting through there, not small molecules, not light. And so you can see is we can still get this activation of SNAC cells that are in these hard tissue mimics. And so, okay, we got this activation of fluorescent proteins, but can we do something a bit more interesting, right? The idea would be that if we want to use these magnetic systems as actual controllable drug delivery devices, we want to be able to release things controllably. And so what we did is we then encoded the pore protein alpha hemolysin on the inactive DNA template. And then we used a model drug molecule, which is just a small molecule fluorophore, to see if we could then release that with the magnetic field. So here you can see the green in the system is now, instead of being a fluorescent protein, is this small molecule fluorophore, and we got this fluorescent dextran again. So if we just leave it with an magnetic field and just leave it as is, obviously nothing comes out, right? The green molecule just stays in there. But if we then put a magnetic field on the system, we get the expression and we get the release, right? So now this is a magnetically activated drug delivery from a SNAC cell. And so you can then measure this, and you can see that this is a bit of a complicated figure, but I go through it. So with the inactive ones, nothing happens. The green molecule stays in. With the full template, we get the release, obviously, that's the positive control. But with the inactive template and the SNAs, without magnetic field, we don't see any release. But with a magnetic field, we see this similar release as if we had the full template, right? So we're getting really great control of the drug release in this using a magnetic field. Okay, so some stuff we haven't published that's like moving on from this is that we can put other things, obviously, onto the nanoparticle to control that activity, because it's just an oligo sequence, right? You can just change that oligo sequence. So going back to what we spoke about at the beginning in terms of an off state, turning an off switch, we've also now just switched the sequence here and put an antisense oligonucleotide therapeutic, right? So in this case, we can think about turning off the translation, but with a magnetic field instead. So the idea would be we have the ASO hybridized to an ASO sequence attached to the nanoparticle, put a magnetic field in the system, which then would release, bind to mRNA, and then if we have RNase H present, degrade the mRNA. And so here you can see how that works, and it works pretty well. So in this case, you've got mRNA, and then again, the control where the ASO cuts it in half, well, not in half, sorry, but cuts it in two. And then with the magnetic SNA, without a magnetic field, you get a really great off state where the mRNA is stable. But when you put the magnetic field in the system, you get this cutting, right? Really great cutting. So you're getting, again, now we're getting this gene knockdown, but in a magnetic field instead of with light or anything else. So we think that's really cool, both for controlling SNAC cells, but also controllable therapeutics of nucleic acids. So I've left lots of time for questions. I thought it'd be good to leave lots of time in this. So just to sort of summarize, we work on oligo conjugates, trying to do a lot of chemistry on oligos and then using those in SNAC cells to get all sorts of different types of application. And in this talk, I spoke all about the remote control. So we use light to get really precise regulation, and we're using now this magnetism to get this potential in vivo control. We're trying to see how far we can take that now. And so hopefully a lot of people with SNAC cells are not chemists. They're in all sorts of different fields, physics, biology. And I just wanted to emphasize that chemistry has a really important potential for SNAC cells by being able to use these synthetic parts and put them into SNAC cells. So with that, thanks to my fantastic group, all these people in blue were people that helped led the work, and then funding and collaborators, and you for your attention, and obviously happy to take any questions. Thanks a lot.
Thank you. I just first I want to say that I really appreciate that you spoke like a chemist. I'm a chemist too, and I'm really glad that this field is seen as more of a chemistry than old-fashioned biology. There's a lot of questions in chat. Can you see the chat?
Oh, not where I am right now. But no.
I can read the questions to you too, if that's easier.
Yeah, I don't know why I can't see them actually, but yeah, if you could, that would be fantastic.
Yeah. Okay, so Donna was asking, this might be naive question. Sorry if this was addressed elsewhere. What is the advantage of one-pot pure over the traditional method? Is it just time saving?
Yeah, I mean, it's a great question, right? There's different things that you're comparing it against, right? So I mean, so Kate, you use lysates, right? And there's different advantages compared to lysates. So compared to lysates, there's not much of a time saving, right? So lysates are also very easy to prepare, right? Just crack open some bacteria and you've basically got it there. The advantage is it's purer, much purer, right? So lysates have a lot of things in them that we don't know, right? We can't really say that using a lysate is understanding everything that's in a SNAC cell, because it's not. Whereas compared to things like the commercial Pure Express or Myfrex or all of these other ones that I don't know the names of now, it's much cheaper, right? Much quicker to do. I mean, if you have to, you can go away and purify 37 proteins individually, right? If you wanted to, right? You can do that. It just takes forever. So to be able to do it this way is much easier. Ribosome purification we've had not much of a problem with, and yeah, Kate's leading on tRNA, which we're all going to need soon enough.
Thank you. Okay, next question. Harvey's asking, this is beautiful work. What % of synthetic cell liposomes do you see burst upon irradiation?
None upon radiation. So like the wavelength we're using is not a wavelength that will activate the lipids. So we'd have to be using much shorter wavelengths to activate lipids in any way.
Okay.
I mean, it's also like, I mean, you may actually have this already. In one of the questions, I always have something about UV damaging something. We don't see damage from UV. So you have to add quite a lot of UV to damage things, right? And so you can do stuff. I mean, we've used these things with living cells as well. So we made light activated plasmids and used them in cells, right? And we had to use a lot extra UV to show that when we were starting to get degradation of cells. Funny enough, I think SNAC cells are probably more susceptible than living cells because they don't have any sort of repair stuff. But we still didn't see any problem with SNAC cells with things like UV.
Yeah. Okay, Bashak is asking, fascinating work. Do you have any light switch specifically for the protein degradation?
Not yet, not yet. That's yeah. I mean, protein degradation's a really important one, and I don't see why we wouldn't, because we could just encode something, right, on a DNA that would degrade things. So I think that's relatively simple. It's about using the right system of degradation. You know, if it's going to be like a ClipX tag, or if it's going to be something like putting a tag in the middle of the protein that you can cleave with a protein cleavage enzyme. I think it'd be relatively simple.
Mhm. Adam is asking, actually, I want to follow up on your previous answer. Please do that, because we badly need it.
Yeah. The protein degradation thing is actually really, yeah, it's you're right. It's a really important area.
All right, Adam's asking, when you have the sender-receiver system and you co-culture synthetic cells with E. coli, do you see that natural bacteria eat synthetic cells, try to consume the liposome filled with proteins?
It's a fantastic question, right? So we specifically incubate them separately in terms of spatially, right? The bacteria is not eating into the agarose, and the SNEC cells are not moving out of the agarose, obviously. There have been people in the literature that have put them together. I don't know exactly how that worked out. I would be surprised if the bacteria didn't disrupt the membranes of the SNEC cells. I mean, I wouldn't think they would eat it. Like I don't think they'll eat the internal contents. It'll just spew out into the extracellular medium. But yeah, I mean, getting that interaction is something that we're quite interested in doing, and I think that would be quite important to do that. I mean, people have done similar things and shown that they can be next to cells. It's not out of the question. It's just they will probably do that over time, and it's how you then reinforce the membrane or things like this, which is some things we're quite interested in doing.
Yeah, Adam, if you want to look up more of that work, look up Sherif Mansi's papers. He's been doing really beautiful stuff in co-culturing.
Yeah.
Okay, Claudia's question.
Abby Schroeder in terms of putting things into mice, right?
Yeah, that's true. That then they do survive. You're right.
And
There's
you see
no.
them in you see them in there. So yeah, they're not all degrading, that's for sure.
Yeah.
And this is just with standard lipid membranes, not even trying to reinforce them in any way.
Yeah. I guess you can't, if you, especially for the stuff that Avi is doing, you need the alpha kim lysine to be able to go into the membrane. So you cannot do any crazy cross-linking of the membrane.
No, but you could potentially do things on the outside of the membrane, not on the inside. But you're absolutely right. You've got to be careful about what you do to the inside of the membrane.
Yeah. Okay, Claudia is asking, tissue mimic is really fascinating. What is the stability of the tissue? How long do you get the synthetic cells to remain translationally active?
So I feel like those are two different questions. So I mean, the tissue mimic is very minorly a mimic, right? It was just a black tube. So like, I'm not gonna big up that too much, right? It was for a very specific purpose of trying to show that we could get through something that cannot get got through by light or small molecules, right? We would love to start doing something on a bit more complicated tissue mimic. I don't see why that wouldn't work. It's more just like getting it set up in the system we have for the magnetic field, right? And otherwise having to buy a new generator of magnetic field, which is obviously grant dependent, right? Sorry, what was the second question? What was the second part of the question?
How long do you get the synthetic cells to remain translationally active?
Okay, so this is a good question, right? Because this question is a whole field question, because the cell-free protein synthesis systems do not last forever, right? And this is just a general problem that no one talks about in the field, right? They when you mix them together, they go, even if you don't have a DNA in them, right? Or if you have an inactive DNA in them, right? So if you leave it for a while before you turn it on, then it won't work, right? If you add a DNA template after a few hours, it won't work anymore. In terms of how long they'll express for from the beginning, again, up to maybe about 6-8 hours, you'll start to get, it'll plateau before then and just slowly go up as long as you're pretty careful about the RNases and things like that.
Yeah. And you're right that we're all called out here because we need to get those systems to work more sustainably, more for longer.
It's all about understanding them more, right? We don't understand them as well as we think we do.
Yeah. Okay, Matt is asking, how long do a double-stranded DNA sequence can you melt off using the magnetic field?
That's a critical question, right? So at the minute, it's just these short oligos. We are in the process of optimizing the nanoparticles to try and get longer oligos being melted off, right? Because obviously, as I said, it's a distant dependent temperature, right? And so it's all going to be about how much temperature we can generate in which location. And so that's something we are actively optimizing right now.
Yeah. And Dan is saying, fantastic talk. What lipid composition are your liposomes?
Yeah, it's normally just PABC, sometimes PABC cholesterol, standard lipid mixes.
As one does.
That's what does.
This is the end of the chat questions. Last call for any questions. I just want to say I really love what you're doing, Mike, because it's not just foundational research, but it also, I can see the path to practical applications, like justifying our existence. And that's super fascinating for the whole field.
With stem cells, right, it's sort of two different routes, right? It's the trying to make them living and trying to use them not living.
Yeah.
Right? And they're both important, right? And they both will learn from each of us about both those things, right? This is what like the thing of understanding what we've even got there is vital for the therapeutic route or whatever application route we're going to use for them.
Totally. And if we want to grow the field, I mean, I'm all for foundational research, but if we want to grow the field, we need the applications to materialize.
That is very true. That is very true.
All right. Thank you so much.
That's all right.
If there are no more questions, I want to say thank you so much for spending morning, evening, afternoon with us, and have a great rest of your day.
Great. Thanks, everybody.
And bye.